Hello, my name is Petra. I'm a senior environmental scientist with the California Department of Water Resources, and I'm here to talk to you about my project, which is about mercury in tidal wetlands. And specifically, I was looking in tidal wetlands in our Sacramento-San Joaquin River Delta and the Suisun Marsh. So why am I studying mercury in tidal wetlands, you ask? Well, the good news. In the last decade or two, DWR and other agencies and other groups have been restoring tidal marsh, including tidal wetlands. And so there's also tens of thousands of more acres of restoration in the works and planning. So tidal habitats is something that's becoming a lot more of an interest to scientists and to the public. But the bad news is California has a really environmentally devastating legacy of gold and mercury mining. And as you can see on this map, we also have a lot of natural mercury that was mined out of the coastal range. You can see those little red dots, and all those yellow dots are gold mines that we had, and approximately there's 47,000 dots on that map. The mercury was carted from the coastal mines, from the coastal mercury mines over to the gold mines, and the gold mines they use the mercury to remove the gold, so there's less gold and more mercury now in the habitat. So what does this have to do with tidal wetlands, you ask? Well, for a long time as scientists, we've been wondering if the more toxic form of mercury, methyl mercury, was being made in these tidal wetlands. Other forms, or other types of wetlands do have methyl mercury, this toxic form being made there. So the more toxic form is a neurotoxic. It's a very potent neurotoxin, and it passes the blood-brain barrier, and it also can be passed from mother to fetus very easily. It's incredibly toxic. The toxic form is also made under very certain conditions. So it's made by bacteria under certain condition, and it ends up in the food chain in high amounts in fish eventually. We eat that fish as humans, or birds eat that fish, or mammals eat that fish, and we're all at the top of the food chain, so it's bioaccumulated up the food chain, and eventually there's a lot more of it for us to get sick with, and it can really, really, really make us sick. So we really don't want this toxic form of mercury to be made by bacteria. In the end, if these tidal wetlands make a lot of this toxic mercury, then we need to know about that, and we need to rethink how we're restoring tidal wetlands and tidal marsh. So that's what my study was about. It was to see if this toxic form of mercury was being exported from these tidal wetlands. How did we measure how much toxic mercury was being exported and imported from these tidal wetlands? Well, we found wetlands that had a closed system, so we were able to measure how much water was going in and going out. They had one or two openings, and we could put a flow meter there. We could measure exactly how much water was going in and out. We couldn't have any leaky wetlands. Then once a month or so, we'd measure the mercury concentrations over a tidal cycle, and we were able to figure out with that flow and that concentration data how much mercury was going in and versus going out. It's an incredibly difficult thing to do, as we're dealing with hundreds of parts per trillion with some of this toxic form of mercury, and it's very challenging, and it's somewhat expensive, which is why we only were able to do it once a month. We measured this for a year at 4 different wetlands, and we looked at the results to look at these snapshots in time. So we had a 25-hour tidal cycle, and we were able to use that data and estimate how much was going in and out versus every month, on a monthly basis. So what did we find? Well, we found that sometimes more toxic mercury did leave some of these tidal wetlands, and sometimes it didn't. Overall, over the year that we studied each tidal wetland, however, we found that none of the 4 tidal wetlands were exporting large amounts of this toxic form of mercury like had been assumed. It's really helpful to know this, because now we know that we're probably not creating large amounts of toxic methyl mercury that's being exported. And it's really helpful to know this, but what would be more helpful is to study more tidal wetlands, and also to look, because we only looked at 4, and also be able to take more sample. As I mentioned, we were only able to take samples about once a month. This is, however, more wetlands that have ever been studied at one time, and this is probably the largest data set of its kind. And for future directions, we would also help to know if those organisms that are living, those little critters that are inside the wetland, have more mercury, or if they have less than places outside. And if they have more, we need to figure that out too. So that's the next steps for doing studying. So my study is just a little piece of this bigger puzzle, as you can see, and I hope you learned something interesting about mercury and tidal wetlands in the Sacramento Valley. Thanks.